Good afternoon. I'm Brother Christopher Turner in the House of Justice in the 11th grade. And with the camera is Brother Angel Siguenza, also in the House of Justice and in the 11th grade. Animal testing is bad. It is harmful to animals. It is abuse that goes unpunished. Animal testing is wasteful, dangerous, and unreliable, and animals are just downright different from humans. Animal testing is wasteful because despite the use of over 115 million animals in experiments globally each year, only 46 new medicines were approved in 2017 by the leading drug regulator, the U.S. Food and Drug Administration. Many of those were for rare diseases. The U.S. drug industry invests $50 billion per year in research, but the approval rate of new drugs is the same as it was 50 years ago. Only 6% of the 4,300 international companies involved in drug development have registered a new drug with the U.S. Food and Drug Administration since 1950. Even those drugs that are approved are not universally effective. Due to individual reactions, the top 10 highest grossing drugs in the U.S. only help between 1 in 4 and 1 in 25 people who take them. Over 1,000 potential stroke treatments have been successful in animal tests. Only approximately 10% progress to human trials. None work sufficiently in humans. A review of 101 high-impact basic science discoveries based on animal experience found that only 5% resulted in approved treatments within 20 years. Animal testing is dangerous. Vioxx, a drug used to treat arthritis, was found to be safe when tested in monkeys and 5 other animal species, but has been estimated to have caused around 320,000 heart attacks and strokes and 140,000 deaths worldwide. Human volunteers testing a new antibody treatment at Northwick Park Hospital, UK, in 2006 suffered a severe allergic reaction and nearly died. Testing on monkeys at 500 times the dose given to volunteers totally failed to predict the dangerous side effects. A recent drug trial in France resulted in the death of 1 volunteer and left 4 others severely brain damaged in 2016. The drug, which was intended to treat a wide range of conditions, including anxiety, Parkinson's disease, was treated in 4 different species of animals, mice, rats, dogs, and monkeys, before being given to humans. A clinical trial of hepatitis B drug, fialdurine, had to be stopped because it caused severe liver damage in 7 patients, 5 of whom died. It had been tested on animals first. Only 1/3 of substances known to cause cancer in humans have been shown to cause cancer in animals. Which leads us to the next point. Animals are different. Animals do not get many of the diseases we do, such as Parkinson's disease, major types of heart attacks, many types of cancer, Alzheimer's disease, HIV, or schizophrenia. An analysis of over 100 mouse cells types found that only 50% of the DNA responsible for regulating genes in mice could be matched with human DNA. The most commonly used species of monkey to test drugs safely, marquis monkeys, are resistant to doses of certain drugs that would be deadly in humans. Due to many important differences between monkeys and humans in brain structure and function, data collected from monkeys used in neuroscience research are misleading and of poor relevance to people. Think about that. Chocolate, grapes, raisins, avocados, and macadamia nuts are harmless to people, but toxic in dogs. But they're supposed to test our medicine. Aspirin is toxic to many animals, including cats, mice, rats, and it would not be on our pharmacy shelves had it been tested only according to the current animal testing standards. Which also shows that animal testing is unreliable. 90% of drugs fail in human trials despite promising results in animal tests, whether on safety grounds or because they don't work. Cancer drugs have the lowest success rate. Only 5% are improved after entering clinical trials, followed by psychiatric drugs, 6% success rate. Heart drugs, 7% success rate, and neurology drugs, 8% success rate. None of those broke 10%. Using dogs, rats, mice, and rabbits to test whether or not a drug will be safe in humans provides little statistically useful insight. The study also revealed that drug tests on monkeys are just as poor as using any other species, predicting the effects on humans. Out of 93 dangerous drug side effects, only 19% could have been predicted by animal tests. Using mice and rats to test the safety of drugs in humans is only accurate 43% of the time. Out of 48 cancer drugs approved by the European Medicines Agency from 2009 to 2013 to treat 68 types of cancer, almost half showed no survival benefits, according to a recent study. Even in cases where benefits were seen, the difference was judged to be clinically insignificant. So think about that next time you hear animal testing is good or it benefits our society. Think about the poor animals and think about how useless that data is to us. Something that's animal tested or tested in animals could end up being your death. Thank you.